The assay represents an important research tool to simulate the degranulation of basophils induced by the cross-linking of patients' IgE and the allergens. Thus, the assay can be used to mimic type 1 allergic reactions.
The assay is robust, reproducible and tailorable. Moreover, it is highly sensitive as it can be performed with small amounts of recombinant or purified allergen or even with complex allergen extracts.
The method is used to diagnose allergies as it analyzes the reactivity of patients' IgE to allergens. Also, this method is suitable to analyze cross-reactivity and to monitor AIT treatment efficacy.
Begin by harvesting Ag8 cells from the cell culture flask and transfer the cells into a centrifugation tube. Pellet down the cells by centrifugation for 5 minutes at 250 times G at room temperature. Aspirate the supernatant and resuspend the cell pellet to a final concentration of approximately 1 times 10 to the 6th cells per milliliter in humanized RBL cells medium. Dilute human sera 1 to 10 in Ag8 cell suspension for the final serum dilution of 1 to 20 in the assay, and incubate for 1 hour at 37 degrees Celsius.